Hello and welcome back to the Biochemistry for Health Sciences channel. Enzyme kinetics is a very important topic in the health sciences. We shall have several videos on enzyme kinetics. This video is an overview of enzyme kinetics. What is enzyme kinetics? Enzyme kinetics is the study of factors that affect an enzyme's function. Remember, the function of an enzyme is to increase the velocity or the rate of a certain chemical reaction. We call this catalysis. Enzymes are catalysts. Let's consider the key steps in enzyme kinetics. Step number 1. You have to know how to determine the initial velocity V of a reaction that is catalyzed by the enzyme. Let's use alkaline phosphatase, ALP, as an example of an enzyme to see how we determine the initial velocity. We can conveniently measure the initial velocity for alkaline phosphatase if we use para-nitrophenyl phosphate or PNPP, para-nitrophenyl phosphate, this compound here, as the organic phosphate substrate. In the presence of the enzyme alkaline phosphatase, this phosphate group is removed as a product, and the other product that we get is this one here that we call para-nitrophenolate or PNP. So PNPP is the substrate, and PNP, which is yellow in color, it is yellowish in color, this is a very convenient product that we can monitor with time. Since this product is yellow in color, we can monitor the production of para-nitrophenol very easily by using a UV visible spectrophotometer. So let's see how we can measure the initial velocity of this reaction that is catalyzed by alkaline phosphatase. So we take a cuvette, and to the cuvette we add buffer as well as a known amount of the substrate, para-nitrophenyl phosphate. We set the temperature. Most enzyme kinetic experiments are done at room temperature 25 Celsius or body temperature 37 Celsius. Now it is very important to maintain the pH, and that's why we need the buffer, as well as the temperature for enzyme catalyzed reactions, since both these factors will change the velocity of the reaction. In the next step, we add a small amount of enzyme, alkaline phosphatase, to the cuvette. Since enzymes are catalysts, we need very, very low concentrations of enzymes. Typically, this will be in the micromolar amount or nanomolar amount compared to the substrate. The substrates typically will be in the millimolar amounts or even in the molar amounts. Once the enzyme is added, the alkaline phosphatase will catalyze the breakdown of the substrate para-nitrophenol phosphate into para-nitrophenol, and since this is yellow in color, the absorbance at 405 nanometers will go up, and you will see the increasing absorbance with time. When you plot the absorbance at 405 nanometers with time, you might see that the absorbance is linear for a few minutes and then curves off as time progresses. This may be due to several reasons. One is that as the substrate is consumed in the reaction, it may become limiting. The second reason is as the product is formed, the product might inhibit the enzyme. And another reason could be enzyme denaturation. So we use the linear or the initial part of this curve to calculate the slope of the line, the delta A per minute. In this example, the line is linear for 3 minutes and the absorbance increased from 0 to 0.1 in 3 minutes. So 0.1 divided by 3 gives us a slope of 0.03. As we discussed in the last video, we can determine the initial velocity, that is, how much para-nitrophenol product is formed per minute, by plugging in the slope delta A per minute, dividing it by the molar extension coefficient as well as the Kcat path length. So this is how we find the initial velocity of an enzyme catalyzed reaction. In the next step of enzyme kinetics, we now determine the initial velocity at various substrate concentrations as keeping the enzyme concentration the same. The enzyme concentration is kept constant, but substrate concentration will change. So let's see how we determine the initial velocity under different substrate concentrations, keeping the enzyme concentration the same. In the first step, we take different cuvettes and we add increasing substrate concentration, P and PP, to the cuvettes. We then set the temperature, once again either 25 degrees Celsius or 37 degrees Celsius. And you can see the concentration of substrate is increasing from cuvette number 1 as we go to cuvette number 2, 3, 4, and 5. We then add a fixed amount of enzyme to all the 5 cuvettes. And then in the third step, we monitor the absorbance with time in each cuvette and we determine the slope or the delta A per minute from the straight line portion of these plots. So from the delta A per minute, we determine the initial velocity. So we have our initial velocity at various substrate concentrations. Once again, keep in mind the concentration of enzyme in all of these cuvettes is fixed. It doesn't change. So only substrate concentration changes and we determine the velocity, the initial velocity at each substrate concentration. So this is the data we need. This is a very important piece of information, important piece of data that we need for enzyme kinetics. So keeping the enzyme concentration fixed, but changing the substrate concentration is quite similar to what we see in healthy cells. At any one time, enzyme concentrations do not change much. However, substrate concentrations can rapidly change. And the reason for this is it takes time to make an enzyme or destroy an enzyme. So enzyme concentrations are more or less quite constant at any given time. However, substrate concentrations can change dramatically. In step 3 of enzyme kinetics, we now plot the initial velocity V of the reaction against the substrate concentration. And if we do this, we typically see two types of curves. One would be the rectangular hyperbolic curve, and the second is the sigmoidal curve. From these curves, we can then obtain three very important values. One is Vmax, the second is Km, or sometimes called S0.5, and the third is Kcat. Making a curve requires several reliable points, and also trying to get these values from curves might require sophisticated statistical software. So another way to get Vmax, Km, and Kcat is to convert these curves into straight lines. So we can mathematically convert the curve into straight lines. We can get the Lineweaver-Burk plot by plotting 1 over V on the y-axis against 1 over S on the x-axis. Or we can get the Eadie-Hofstee plot by plotting the initial velocity V on the y-axis and on the x-axis V divided by S. Or we can generate the Haynes-Wolff plot by plotting S divided by V on the y-axis against S on the x-axis. And it might be much easier to get Km and Vmax from these straight lines. So once we can determine Vmax, Km, and Kcat values, these values will help us answer many important types of questions in biochemistry as well as in the medical sciences. So examples of these questions include how good is an enzyme? Is this a perfect enzyme? Why does my face become red after I drink a couple of drinks? Or will I become an alcoholic? How do enzymes change in diseases such as breast cancer? How do enzymes help us diagnose diseases? How is metabolism regulated by enzymes? How do drugs such as statins reduce blood cholesterol levels? We will discuss these important features on enzymes in the next few videos. Thank you for watching. Goodbye and have a wonderful day. And please do not forget to subscribe. It is free.